Hello everyone, I'm Dr. Arpita. A very warm welcome to all of you in my developmental biology classes. The topic for today's discussion is patterns and types of cleavage. You can see this topic. This is one of the most important topics in your developmental biology. And let me tell you, once you are confident with this topic, once you are clear with this particular topic, you can understand all the subsequent topics that are going to come. Okay, so let's get started. The term cleave means to split, to cut and split. Okay, see, it means to split or slice apart an entity. It need not be an egg or any biological substance or anything like that. It is simply splitting something apart. It is the process by which cellular division occurs during the zygotic phase of development that ends with a multicellular embryo. This is what you are getting. Okay, it is ending. The phase ends with a multicellular embryo. You see, this is being split, cleaved subsequently. And what you get finally is a multicellular embryo, right? The see a zygote is what? You all know that by now. A zygote is the fertilized egg. Okay. See, the egg gets fertilized by the sperm cell to give rise to the first cell of the next generation, that is zygote. Okay, so a zygote is a fertilized egg that is ready to begin many rounds of incredibly fast cellular division. The process of DNA cleavage consists of cellular division without the growth or differentiation of cell. This is a very important line. You should all take a note of it. The process of DNA cleavage consists of cellular division without growth or differentiation of cell. Growth and differentiation of cells will come later. It will not be a simultaneous event that growth as well as differentiation of cell, everything will not take place along with this DNA cleavage. It is something that is going to follow DNA cleavage. Correct? See, cellular division is associated with DNA cleavage, but mind it, it is not associated with growth and differentiation of cell. Please take a note of this. The process of DNA cleavage consists of cellular division without the growth or differentiation of cells, right? Cleavage ends when the embryo becomes a blastula. So when it is becoming, when cleavage ends, okay, you know that the embryo has become a blastula. And that's what, or a hollow ball of cells via a series of rapid mitotic divisions. So end of cleavage is marked with the formation of blastula, which is a hollow ball of cells, and that is formed via a series of rapid mitotic division. Clear? Now we'll come to the properties. The two most important properties, see, what is the first one? Rapid cell division without any pause through mitosis. Okay, here the next one, total volume may be fixed, but size of the cell decreases. See, here I'm doing this briefly so that during the time of exam, you have enough load on your head. Normally you have that. At that time, if you just go through it once, it will be ready for you. Hmm. See, once fertilization takes place and a new organism begins to develop, what happens that time? You just see the zygote. Okay, the zygote begins to cleave. Clear? The zygote begins to cleave. The site of beginning of cleavage is known as cleavage furrow. So what is cleavage furrow? It is the site of beginning of cleavage is known as cleavage furrow. Clear? The zygote begins a series of very fast mitosis-like division, but instead of creating cells the same size and growing, the cell division results in much smaller cells. So what do you understand here? See, read it nicely. See, what does it say? The zygote begins a series of very fast mitotic division, mitosis-like division. Clear? But instead of creating cells the same size and growing, the cell divisions result in much smaller cell size. So the zygote doesn't experience growth during cleavage. See, if the cells are also growing bigger, this means what? The zygote will experience growth, but the zygote doesn't do so. The resulting blastula ends up the same size as the zygote was before cleavage had commenced. Very important line. See. Since there is no growth, what's happening? The resulting blastula ends up the same size as the zygote was before cleavage had commenced. Factors that can regulate the development of cleavage include the amount of egg as well as the distribution of yolk. Fine. Now we should know few laws of cleavage. First law of cleavage is which one? See, the Balfour's law. What does it say? The rate of cleavage is inversely proportional to the amount of yolk. That means what? If the amount of yolk is more, the rate of cleavage will be less. And if the amount of yolk is less, the rate of cleavage will be more, because the yolk creates a hindrance to cleavage. Fine. Now, this one can be written in easiest form. In which way? The rate of cleavage, I said, is inversely proportional to the amount of yolk. That means what? More yolk means slow rate of cleavage. Less yolk means high rate of cleavage. Correct? Next, what is that? Hardwick's law. What does it say? The axis of mitotic spindle occupies the longest axis of protoplasm, and cleavage furrow tends to cut it transversely. So what is it? The axis of the spindle has an axis, right? The axis of the spindle occupies the longest axis of the protoplasm, and the cleavage furrow tends to cut it transversely, like this. It tends to cut it transversely. And the last one, what is it? Sachs law. Cells tend to divide into equal size of daughter cells. Cells tend to divide into equal size of daughter cells, and each plane of division tends to bisect the previous plane at right angles. Remember this. Okay, each plane of division tends to bisect the previous at right angle. That means so if the previous one is like this, the next one will be like this. See, an angle of 90 degrees formed here. Got it? Okay. Next is patterns of cleavage. Clear? Next, we are going to talk about patterns of cleavage. Cleavage is broadly classified into two big heads. What is that? Holoblastic cleavage and meroblastic cleavage. Okay, just for your reference, take a note of these two diagrams. This is holoblastic cleavage and this is meroblastic cleavage. What happens here? Let's see. The pattern of embryonic cleavage particular to a species is determined by two major parameters. So the pattern of embryonic cleavage is actually determined by two major parameters. What are they? The amount and distribution of yolk protein within the cytoplasm, as we just now have discussed, that if the yolk is more, cleavage becomes slower, and if the yolk is less, cleavage becomes faster. And next is what? Factors in the egg cytoplasm that influence the angle of the mitotic spindle. Okay, it influences the angle of mitotic spindle and the timing of its formation. So there are certain factors in the egg cytoplasm that influence the angle of mitotic spindle. At what angle will it be? Angle of mitotic spindle and timing of its formation. Is that clear? Next, coming to, straight away we'll go to holoblastic and meroblastic cleavage. So what is holoblastic cleavage? Holoblastic cleavage is that type of cleavage where you can see that the cleavage plane is dividing the zygote as well as the blastomere completely. So you just remember holoblastic is associated with the word complete. Clear? It is associated with the word complete. So if the cleavage plane divides the zygote and blastomeres completely, it is known as holoblastic cleavage. Then what is our meroblastic cleavage? In the presence of large amount of yolk, take a note, in the presence of large amount of yolk in the fertilized egg or egg cell, the cleavage planes divide the zygote and blastomeres partially. Okay, the division is not complete. It is known as meroblastic cleavage. Clear? Now see. Now we will discuss these in details. I hope my voice is clear. You can hear me properly. If not, please feel free to say. Okay. So we'll start with holoblastic cleavage. See, I'll write it down. Please take a note of it. And as I always say, try to understand it. Don't just blindly sit and copy. Clear? Should we start? Okay. First, take a few seconds to study the diagram. What can you see here? 1 becomes 2. 2 becomes 4. 4 becomes 8. 1 becomes 2, 2 becomes 4, 4 becomes 8, like this it is going. Yes or no? Okay. So, our first point, what should we write for our reference? Holoblastic cleavage means complete cleavage. Clear? See, in isolecithal eggs, first thing we'll talk about isolecithal eggs. What are isolecithal eggs? I have discussed this with you earlier. What happens in isolecithal eggs? What are isolecithal eggs? Hmm? The, just write in bracket, yolk is less sparse and evenly distributed. Agree? Yes or no? Please say something. See, these types of eggs and all, I'm not discussing in detail. You know it. So, just for recalling purpose, I've written here isolecithal eggs are eggs with lesser amount of yolk and they are evenly distributed. Clear? Okay. Next, what we should know here, see, I'm writing. This is your radial cleavage, which is found in, I should write here. This is radial cleavage, which is found in echinoderms. Then it is found in Amphioxus. Just no bullet points. Don't have to write Ramayana, Mahabharata, epic stories here. Okay. So what have you learned here? See, you have learned that isolecithal eggs are eggs with lesser evenly distributed yolk. This is radial cleavage. See, this pattern of cleavage is radial cleavage. Okay, this radial cleavage is found in echinoderms, Amphioxus, okay. Next, see, next we are talking about spiral cleavage. This is, all these happen in isolecithal eggs. This is 1, this is 2, this is spiral. In spiral, see, you will have to be very particular about the way in which the cleavage is taking place. The plane of cleavage has to be studied with a lot of attention here. In spiral cleavage, this pattern is followed. The pattern is very, very important. If you remember, see, we had talked about patterns of cleavage here. Okay. So spiral cleavage is found in annelids, then mollusks, then flatworms also. Hmm? Got it? Next, this one is what? This is your bilateral cleavage. Hmm. Remember, this bilateral cleavage is found in tunicates. Very, very important. See, it is cutting bilaterally. Hmm. It is found in tunicates. Next, what do we have? Hmm. This one, here you can see. This is the fourth one. Fourth one is rotational. This is, I've seen in different exams, this comes. The rotational cleavage is very important. Why? Because it is found in mammals. Please take a note. Hmm? This is found in mammals also. It is found in nematodes. Hmm? Now, what are these? I'm not discussing in details. What is mm, the spiral? What is rotational? All these have already been discussed. You know these. This one I told you. It's the revision class. So I'm not going into details. Just the bullet points I'm highlighting so that it is ready during your exam. What is this one? Hmm? Hmm? What is this? I would expect you to answer this. This is B. This is a hint. See, this one is A, isoleucical. This one is what? Mesoleucical. Mesoleucical. Fine. Is that clear? This is mesoleucical. In mesoleucical, what is this? This is associated with moderate amount of yolk. Hmm. Moderate amount of yolk. Vegetal disposition. Very important. And what happens here? See, this is also a type of radial cleavage only. See, this one is little similar to this one. Huh? This is also radial. Radial and mesolithical eggs, and it is found in amphibians. Important, huh? Amphibians, very, very important. Next, we'll go to midoblastic cleavage. You have already learnt about holoblastic cleavage. This is your holoblastic cleavage where we have two types of eggs, isoleucical, mesoleucical, and we have four types of cleavage, radial, spiral, bilateral, and uh, this rotational. Okay. And for each case, you have got examples also, which you should not forget. Next, we will talk about midoblastic or incomplete cleavage. Midoblastic or incomplete cleavage. Now, what happens here you see for the first one? Telolicital eggs. In telolicital eggs, what happens? Hmm? The yolk is dense throughout the cell. It's not sparsely distributed or anything like that. It is very dense and it is throughout the cell. Clear? Telolicital dense yolk. Got it? Hmm? Fine. This is your telolicital egg. All right. See, in after telolicital, in this telolicital egg, you have this two types again, bilateral and discoidal. This one is your bilateral. This is your discoidal. Okay, bilateral and discoidal. Remember, bilateral is found in the cephalopods, mollusks, all these, huh? Mollusks, mollusks, or especially the cephalopod or cephalopod mollusks. And discoidal is found in, important again, note it, reptiles, fish, reptiles, fish, birds. Okay, discoidal is found in birds, fish, reptiles, amphibians. In mesolithical, you get amphibians, which is very much similar to this radial cleavage. Should know this. Radial. In mammals, it is rotational. Hmm? These terms, you should, the moment you hear this, this term should flash in your mind. Now coming to the last one. This one in miroblastic, A was your telolicital, now B is your centrolicital. In centrolicital egg, what happens? The yolk is in the center. Yolk in the center. Okay. What happens here? This type of cleavage is superficial and it is found in seconds. This is also important. I've seen this coming. Hmm? This is also important. Got it? Fine. Next, we'll talk about the patterns of cleavage. Hmm? See, an egg can be divided from different planes during cleavage. Hmm? Whether you are cutting it from the vertical plane, like this, horizontal. So, egg can be divided from different planes during cleavage. What happens depending on the position of the cleavage furrow, the planes of cleavage are named. So planes of cleavage are named depending on what the cleavage furrow. First is the meridional plane, second is the vertical plane. In meridional plane, what happens is the plane of cleavage lies in The plane of cleavage lies on the animal vegetal axis. In meridional cleavage, what happens? The plane of cleavage lies on the animal vegetal axis, and what does it do? It bisects both the poles of the egg. Bisects both the poles of the egg. Thus, the egg is divided into two equal halves. This is your egg. First half, second half. The egg is divided into two equal halves. Okay. Next is the vertical plane. What happens here? The cleavage furrows may lie on either side of the meridional plane. The furrows pass from animal to vegetal pole. Okay, the furrow passes from where animal to vegetal pole, and the cleavage cells may be unequal in the cleaved cells are not equal. Here see, the cleaved cells are equal. Cleaved cells are of equal size. Here cleaved cells are of unequal size. Next is what? The equatorial plane, and then last is latitudinal plane. In equatorial plane, what happens? This cleavage plane bisects the egg at right angle to the main axis. It lies on the equatorial plane and it divides the egg into two halves. Here also it is dividing it into two halves. And in latitudinal plane, what's happening? It is similar to the equatorial plane, but it lies on either side of the equator. Okay, it is also called transverse or horizontal cleavage. The other name for latitudinal cleavage is what? Transverse or horizontal cleavage. That I hope it is for understanding. There shouldn't be any. That's nice. I hope you are understanding the context, understanding the content of the lecture. That is very important. Huh? Now we'll talk about the significance. Okay, now we'll talk about significance of cleavage. Important, please note. The cleavage converts a unicellular zygote. Yes, note. I've seen many students writing an unicellular. Please take a note of your English, okay? Don't make this mistake. It's a university, a unicellular, and an umbrella. See, I've not come here to teach you English. I'm not getting into the details of it, but please note these important. A university. Don't write an unicellular. This is wrong. Fine? This is wrong. This is right. Yes, yes. Yeah, I understand. Let's not get into English now. I'm not sitting here to deliver English language or grammar, English grammar classes. So you better pay attention to Dev Bio now. We'll talk about that later. After your exam, we'll talk about all these things. Fine? Okay. The cleavage converts a unicellular zygote into a multicellular embryo. So what is happening? See, unicellular embryo is becoming a multicellular embryo. It produces a large number of cells or blastomeres that are needed. See, it is producing quite a large number of cells, right? And it is needed for the building of the baby's body. See, when the sperm fertilizes the egg, it's just one diploid cell. From one cell, and when the baby is born, think of the size of the baby. Such a big baby is born. so many cells in their body, organs, tissues, whole lot of it. How is it happening? This process? Hmm? Fine. The size of the cell and the ratio of nucleocytoplasma is the cleavage. Fine. During cleavage, the blastomeres go through quick mitotic division, after which there is no growth of blastomeres. Correct? Cleavage facilitates the distribution of cytoplasm among the blastomeres. Clear? Okay, so this is just, we are almost at the end of our lecture. This is just a very brief mechanism of cleavage. What happens here, you see? Cleavage is the result of two coordinated processes. What is that? Karyokinesis and cytokinesis. The mitotic division that divides the nucleus, you know, karyon means nucleus, right? Means what? Nucleus. So division of nucleus is karyokinesis, division of cytoplasm is cytokinesis. Okay. So karyokinesis and cytokinesis together gives you cleavage. Fine. The mechanical agent of karyokinesis is the mitotic spindle with its microtubules composed of tubulin, the same type of protein that makes up the sperm flagellum. Clear? The mechanical agent of cytokinesis is the contractile ring of microfilaments. It's the contractile ring of microfilaments made up of actin, the same type of protein that extends the cell microvilli and the sperm acrosomal process. Clear? Okay. So finally, we have come to the end of our lecture. Hope you have liked it. We are soon going to have our discussion class on cleavage and gastrulation in amphibians and birds. That means frog and chick. Okay. Till then, happy learning. Please keep this in practice. Okay. Till then, bye-bye.